Next, I want to bring on a very distinguished emeritus professor, Dr. Don Huber, who has worked in the field of agriculture for 60 years. He has also been an Army colonel. He's traveled around the world. He was recently advising the government of Ukraine on agricultural issues. But he is best known for identifying problems with glyphosate and solving those problems. He has an enormous research background, and I guess I'm just going to let him tell you more about that. He knows more about glyphosate than probably anyone in the world. And so everybody needs to listen carefully. Thank you, Dr. Huber.
Thank you for the opportunity to participate. We could have the first slide there. I don't see any of there we go. Agriculture is really the basic infrastructure of any society. We have the objective and the responsibility to produce a safe, affordable, nutritious food and other commodities. Next slide. But we need tools to do this. And we use cultural, chemical, and biological tools to produce that food, and it should be safe and abundant for us. One of those widely used tools is an herbicide that we refer to as glyphosate, or common name being Roundup. It was first identified and synthesized as a synthetic amino acid, 1957. In 1964, it was identified as a mineral chelator by Stauffer Chemical Company, patented it, used to clean steam pipes and boilers because it could, by chelating, it grabbed onto the mineral and was able to remove it, descale the boilers. 10 years later, Monsanto patented it as a very broad spectrum herbicide used to kill all vegetation initially, or thought to do that. And in 2002, Monsanto patented as a very broad spectrum antibiotic. It's an antibiotic against beneficial organisms and a few parasites, but it's a stimulus for many of our pathogens. As a synthetic amino acid, it's also very persistent in the environment. Many soils, there's essentially no degradation at all, so that there's been an accumulation for 30 to 50 years of this very unique compound that we refer to as glyphosate. Next slide. The EPA said that it's toxic or inhibitory only to the shikimate pathway, which mammals don't contain in their own cells. But all of the organisms that we rely on in our GI tract and in the environment, essentially, do have this pathway, and yet there have been very few or any. In fact, there are no long-term safety studies that show safety. The only one that is a long-term study actually shows just the opposite. And there are numerous studies, scientifically peer-reviewed studies, independent, that show carcinogenicity, kidney damage, liver damage, intestinal toxicity, neurotoxicity, autoimmune relationships, and reproductive toxicity. We were told that it's very transient, poof, and it's gone. In reality, we know that it can bioaccumulate, not only in our bodies, our brain, our heart, livers, all of our organs and muscle and bones, but also in the environment. As I indicated there, as a synthetic amino acid, there are few organisms that can really degrade it fully. And we were also told that it'll always be very effective. We won't have to worry about resistant weeds, and yet we have numerous weeds that are now resistant to it, so that our input costs for agriculture for weed control are anywhere from $50 to $150 more expensive than they were without the use of the glyphosate. Next slide. Again, this is the shikimate pathway, and they said it only inhibits the EPSPS enzyme, which is right up there at the top. But as a mineral chelator, it chelates the manganese to inhibit the EPSPS. But you have cobalt up there that it also chelates, and it chelates copper, and it chelates manganese, chelates iron. These are all essential minerals for people and animals also. And you can see that all throughout this pathway, it inhibits its individual physiologic pathways as well as shutting down the whole system. So next slide. In reality, it's been recognized that there are actually 291 enzymes that glyphosate can downregulate or inhibit. The inhibition of EPSPS down here is only one of those enzymes. Here's the cobalt up here with the acetoheptulose, and you see all of the other enzymes. Many of these are very critical enzymes for our own physiology, so that you can see that there are many diseases that can be traced back directly to glyphosate. Next slide. Again, we use a horrendous amount of glyphosate. We use about 280 million pounds of glyphosate now for agriculture, and we use another 250 million pounds every year in forestry and other uses, roundup or utility right-of-ways and other activities. And if you look at it as an antibiotic, recognize that antibiotic potential, total indiscriminate application of this antibiotic is almost 500 million pounds of indiscriminately applied by air, by water, and soil, so that the entire environment is exposed to it and almost drenched in it. As an antibiotic, when an organism develops resistance to glyphosate, it has also generally lost its sensitivity to all of our other regular antibiotics, penicillin, tetracycline, cephalosporins, and all of those. Then those organisms essentially become Mars-type organisms. Next slide. Again, its persistence. This is a field in Georgia of pumpkins planted perpendicular to a previous crop of Roundup Ready corn. You can see the effect of the residual activity from the root exudates where the Roundup goes into the soil. And you can see the damage then to the pumpkins from that residual effect. Next slide. You see it also, this is an area where the farmer cleaned out his spray boom or his spray nozzles here in the soil. It's still essentially a barren area for crop production 4 years afterwards because of the persistence of the glyphosate. Next. Again, we can, as we analyze our soils, this is kind of a composite of many soil samples throughout Australia. You look and you can see that as the soil becomes heavier, the glyphosate residue intensifies, so that you see down here 4,155 parts per billion glyphosate in the top 10 inches of the soil. That's essentially 20 years accumulation of glyphosate with very little less than 10% degradation to the AMPA stage, but are in the deeper areas, but very little degradation into AMPA, which is just a single decarboxylation with 1.5 times the toxicity of the initial glyphosate. So we have very large quantities in many of our soils. Next slide. So that we can see the effect of that now, because phosphorus fertilizers and glyphosate have the same binding site in soil. This is a situation in Canada that where they were fertilizing to get 56 bushel per acre of lentils. Their average had been around 26, and for 3 years had been reaching close to the 56, and they wanted to increase their phosphorus availability for the plant and made a foliar application. Then in June 16th, not realizing the elephant in the room was that residual glyphosate in the soil, about 300 or 600 parts per billion glyphosate residue in the soil, applied enough phosphorus to bring them up for that 56 bushel. And then in 3 days, the lentils turned from beautiful green to a light green. And in 10 days, the crop was dead. They harvested 7 bushel and wanted to know what happened to the other potential 49 bushel. Well, when we did the analysis then on the straw from the lentils, had 300 and some parts per billion glyphosate in the lentils, which is more than enough from an herbicidal standpoint to give complete kill. So that we have a lot of our soils now that we're going to be limited on fertility, have to be much more careful, because it can come back to shoot us in the foot by desorbing that residual glyphosate that's been accumulating for many years, especially in our clay soils. Next slide. Again, there are many opportunities for exposure mentioned from contaminated soil. There's a lot in many of our water systems, our lakes and our streams. You look at the U.S. Geological Survey study of glyphosate, there are a few areas that aren't contaminated, and many of those at very extreme rates. So there's many opportunities for exposure that we normally wouldn't think of, such as our school grounds and parks and recreation areas. Next slide. Had an interesting situation 2 years ago where a company making sauerkraut had about $1 million worth of their sauerkraut that came out more like applesauce than it did the good quality sauerkraut that they had normally produced. And it all went into the landfill because it wasn't a commercial product. And trying to find out what the cause of that was, we finally traced it down to contaminated manure that was used as a nutrient source for the cabbage, and the glyphosate came from the feed that the poultry were fed, came through into the manure, contaminated the soil and the cabbage. Then when it was fermented, it blocked the shikimate pathway, tied up the minerals and blocked the shikimate pathway with the glyphosate, so that the secondary cell walls didn't form and it mushed down in the fermentation process. again, this situation with $1 million loss because of soggy sauerkraut and very serious problem from a production standpoint and quality. Next slide. Again, glyphosate is a very powerful antibiotic, but it's an antibiotic against the beneficial organisms for biological control, for mineralization, for soil aggregation, for hormone production, for our plant growth, stimulation. All of those are very sensitive to glyphosate, and it's very toxic to them. However, our pathogens, it's a stimulus too. They're resistant to glyphosate. Pathogens such as Phytophthora, Rhizoctonia, and Fusarium, all of those are stimulated, as Dr. Bob Kramer shows, over 500-fold increase in Fusarium infection of Roundup Ready soybeans. There's nothing in the genetic engineering for tolerance to glyphosate that does anything to the glyphosate. It's still a very powerful antibiotic. It's a very powerful mineral chelator. And so we have very few voids in nature, and the void that's created by taking out the beneficial organisms then is stimulus to the pathogens, and we see the increase in food recalls and contaminated foods, because many of those natural biological controls that would normally contain our pathogens are no longer there as a result of the toxicity of glyphosate that is almost universally or ubiquitous in the environment now. Next slide. As a result of this increase in and stimulation of our pathogens, we have over 40 diseases, plant diseases, that we had previously managed quite effectively and economically effectively with their cultural and other chemical controls and biological controls that now we're referring to as re-emerging diseases. We also have some new diseases that didn't exist before glyphosate because of that disruption of the mineral nutrition and reduction in mineral availability. Next slide. One of those is sudden death syndrome. This disease has cost farmers as much as $1 billion a year. You can see the effect of the glyphosate here in stimulating that disease by the green islands that occur around the power poles. This half-mile long field, and you can, around each one of those power poles, you have green, healthy soybean plants that are still capturing the sun's energy, storing it as sugar and as protein for our consumption, because the farmer pulled away from the power pole enough to make sure that he didn't hang up on the power pole and damage his equipment, so that these beans in the green islands didn't get that treatment with the soybeans that the rest of the field did. And you see the impact of this very serious disease we refer to as sudden death syndrome. Next slide. There are a lot of food and feed safety concerns with glyphosate. Mentioned the reduced nutrient density. Well, as Isaiah said 800 years BC, that all flesh is grass. That's plants are the source of our minerals. And as glyphosate has tied up many of the minerals, in my research, I couldn't find any release of those minerals until the tissue was completely decomposed. And then they would eventually become available, but not available while they're chelated with the glyphosate. We have an increased level of toxic products. The mycotoxins, DON and zearalenone, and the aflatoxins are all stimulated and more severe in the glyphosate-treated plants, whether they be GMO or the residual glyphosate effects on them than they were without the glyphosate. Premature aging and reproductive failure and ecological disruption for bees and amphibians and other beneficial soil organisms. We have direct toxicity also of glyphosate. Next slide. It's not just for cancer. Cancer's one, but there's about 32 of our diseases for humans that are approaching epidemic proportions in our society that are all increased by glyphosate residues that we get through our food or the environment. You can see these are all peer-reviewed scientific studies showing at the extremely low level of glyphosate at the effect on those particular diseases. Next slide. And so as Stephanie Seneff and others will indicate some of those pathways and the effects of glyphosate. But again, it's a very powerful antibiotic. We're indiscriminately applying them in the environment so that we have a complete change in the biological microbiome in these systems. This is a study by Clare and another by Shihata and Kruger showing that antibiotic activity against beneficial organisms here that will suppress the pathogens in our general environment. But the pathogens that are resistant to glyphosate then have a free rein for development to contaminate our food and our feed products and us directly. So that you see the food recalls for Salmonella and Clostridium and Escherichia coli and other pathogens that are not sensitive to glyphosate then occupy that area, and we have the consequences then through increased food recalls and increased disease. Next slide. Again, that powerful antibiotic effect is manifest in many different ways. Dr. Monica Kruger in Leipzig University, Germany, showed that at 1/10 of a part per million in feed, which is a very low level of glyphosate, actually from what we're finding in many of our feed and food products, as Zen Honeycutt indicated, that level, 1/10 of a part per million, is toxic to the Enterococcus. You can see over here in the healthy stomach, the population of the Enterococcus. This is the biological control for Clostridium botulinum. 1/10 of a part per million is toxic to those organisms, so they're not present in the rumen of the animal. Consequently, Clostridium botulinum develops, grows, and produces a bot neurotoxin until the animals are finally killed and finally die. We have the chronic botulism. Many of our large dairies will see as many as 6 cows die a week from this glyphosate toxicity. Next slide. Again, humans aren't discriminated against in toxicity effects of glyphosate. This is a slide of the children that had celiac disease serious enough for hospitalizations. And you can see the epidemiological curve and the increase of celiac as soon as GMO canola and that 5-fold increase in the application of glyphosate on this disease and its increase. Next slide. We see that same curve with these other 32 diseases of humans that we see with the celiac disease with the children. So we see that very serious broad spectrum effect from the antibiotic activity of glyphosate, as well as from the mineral chelation effects to shut down many of our physiologic pathways. Next slide. So that in the past, we've recognized that we have biological accumulation of glyphosate in our soils. We've recommended capitalizing on chelation, getting more positively charged ions, gypsum as a calcium source, or limestone, getting it right down the seed furrow to tie up much of that glyphosate, trying to entrap the glyphosate, keep it inactive from an herbicidal standpoint. And next slide. The other approach, more recently, we've been able to go to degradation, but in tying it up, you can see the effect of residual glyphosate. This is as they increase the, as Crady increased the amount of gypsum that was applied, and you see the health of the root system here and the health on the plant by tying up the residual glyphosate. Next slide. When we tie up the residual glyphosate, we merely kick the can down the road because it accumulates in the soil, and then we have the desorption problem that comes into play there. More recently now, and this is one of the things from the soggy sauerkraut incident, Monica Kruger had shown that sauerkraut juice, raw sauerkraut juice, can desorb, or not desorb, it can degrade glyphosate, the Lactobacillus plantarum that does the fermenting in sauerkraut. It's a good probiotic, but it's able to degrade glyphosate completely. There's also an 8-organism biococktail that Paleobiotica Company has put together that will degrade soil residual glyphosate all the way through the 3 primary pathways that are involved there. And in our study with planting corn a year after the application of raw sauerkraut juice or the Paleo Grow product, you can see that we increase the yield and of the grain yield, also silage yield, and when we combine the 2 of them to get more thorough degradation on it, you can see a 10.8 bushel increase in yield, grain yield, and a 2.5 ton more silage yield in that, just by getting rid of the residual glyphosate and freeing up some of those chelated minerals, especially the micronutrients that are so essential to disease resistance and general growth. Next slide. So in summary, you can't see it or taste it or smell it, but glyphosate can slowly kill you. Next slide. I think that in summary, future historians may well look back and write about our time, not about how many pounds of pesticide we did or did not apply, but about how willing we are to sacrifice our children and jeopardize future generations with this massive use of glyphosate that's based on false promises and flawed science, just to benefit the bottom line of a commercial enterprise. Next slide. To summarize, Monsanto was right in their summarization that Roundup or glyphosate is the ultimate killing machine. Thanks for the opportunity to share. I hope that people will take this to heart and be very serious in the legislation that's coming up, and also in being more observant of contamination in our food supply. Many thanks.
Thank you very much, Professor Huber. That was a fabulous talk. And for the audience, I just remind you that glyphosate, remember his octopus in the beginning, it has so many different harmful effects, and they act synergistically. So that's a problem. And it doesn't only kill off the important microbiota in the soil, but also the microbiome in our gut and in the gut of the cows and the other animals we're eating. So the effects are widespread, very concerning, and the science has been faked by Monsanto and Bayer. We can't really trust it. Dr. Huber can pick out maybe some of the good studies, but it's hard to know what to rely on. Um, it's important to avoid this product.